Hello, everyone, and welcome to today's webinar, Implications of Human Microbiome Research for Epidemiology and Public Health with Dr. Betsy Foxman. This webinar is part of the Summer Omics Learning Series presented by the Omics and Bioinformatics Core of our M-LEEaD Center at the University of Michigan. The event will begin shortly, and as participants are still joining, I'd like to provide a few technical reminders to the group. All participants are muted upon entry and for the duration of the event. This webinar will be recorded and will be made available on the M-LEEaD website following the event. If you have any questions, please submit them to the Q&A box at the bottom of your screen at any time during the presentation. And with that, I'd like to thank you all for joining us today. And I will hand it over to Dr. Maureen Sarter, the lead of the OBIC core.
Thanks, Katie. And good morning, everyone, and welcome to the first Zoom seminar of our Summer Omics Learning Seminar Series. As Katie said, I know we have some new people joining in now. This seminar series being hosted by the M-LEEaD Center of the University of Michigan. M-LEEaD is the Michigan Lifestage Environmental Exposures and Disease Center. So I'm Maureen Sarter. I'm lead of the Omics and Bioinformatics core for our center. And I am extremely pleased to be able to introduce Dr. Betsy Foxman to you today. She is an endowed professor of epidemiology and director of several things with long names. So let's see. She is director of the Center for Molecular and Clinical Epidemiology of Infectious Diseases, director of the Integrated Training in Microbial Systems Program, and director of the Certificate Program in Healthcare Associated Infection Prevention and Control. So Dr. Foxman originally came to Michigan from California back in 1984, beginning her faculty position as a visiting assistant professor, and then never leaving, presumably after realizing what a great place Michigan is. She studies the molecular epidemiology of infectious diseases, and her recent research focuses on how the human microbiota interacts with pathogens to lead to numerous different types of infections and public health issues, including dental health, antibiotic-resistant bacterial infections, recurring UTIs, and preterm birth. Today, she will be speaking on implications of human microbiome research for epidemiology and public health. And as you can see, there are two other seminars in this series coming up on epigenomics and the exposomics. So for now, thank you so much, Betsy, for presenting with us today, and I'll turn it over to you now. Thank you.
Thank you, Maureen, both for the invitation and the kind introductions, and Katie for making all the arrangements. If you all would feel free to please post your questions on the Q&A. Katie will be monitoring the questions and let me know so I can answer them in real time. It's a real pleasure to share with you some of my thoughts on the human microbiome and why epidemiologic research on the human microbiome is important for public health. So I'm going to begin with this little limerick by Hilaire Belloc. The microbe is so very small, you cannot take him out at all. But in fact, microbes are found in all environments, from the Arctic Circle to thermal hot springs, and microbes are present in all plants and animals. It's estimated that humans have 10 times as many microbial genes as human genes, so in reality, we take microbes with us everywhere. Every human body system depends on microbes, from your nose to your colon. At a minimum, microbes prevent invasion by pathogens, a function called pathogen or colonization resistance. But as you can see from this list here, microbes aid in digestion, they fortify the immune system, and they lubricate tissues. If you took biology a while ago, you would have learned that there are commensal microbes living in and on your body that were probably called flora or microflora. But microbes are not plants, they're not flora, so taking that into account, we now use the term microbiota. And the human microbiota has microbes from several different kingdoms: bacteria, archaea, virus, and also members of the eukaryotes, fungi, and parasites. Early investigators first learned about microbiota using the microscope, and then by growing individual microbes in pure culture and studying them in the laboratory. Now using polymerase chain reaction, next generation sequencing, and mass spectrometry, we can obtain a snapshot of the entire microbial community: who's there, what they're doing, what they're capable of doing, and how they interact with their host and environment. We can also visualize the community in vitro and glimpse it in vivo. This has profoundly changed our understanding of our microbiota. And because so much of this research depends on genetic sequencing, it's become common practice to refer to microbiota as the microbiome, which refers to the genome of the microbiota. To date, most studies have focused on bacteria, and often microbiome studies address only the bacteria present, or the bacteriome. These images reflect the differences in technical approaches with time. On the left, you can see a drawing by Robert Koch of Koch's postulates, who drew by hand the anthrax bacillus at various stages of development. On the right, we see an image of dental plaque developed by synthesizing results of high-throughput sequencing data with spatial and structural information by the group of Jessica Mark Welsh. The microbes in dental plaque are not unique in growing these beautiful complex structures or biofilms, because most microbiota live in biofilms, either of single or multiple species, in the environment and in our body. Microbiota live in dynamic ecological communities specific to their local environment. A major area of research is characterizing the interactions within each community and how a community interacts with its host and the external environment. Studies to understand these underlying mechanisms are best done under controlled experimental conditions, either in vitro or using animal models. But to identify which experiments are most likely to result in findings useful to human health requires epidemiologic studies. This figure is from a 1954 paper on epidemiology by Dr. Thomas Francis, the founder of my department and a Lasker Award winner. And it beautifully shows the various contributions of epidemiology to medicine, pathogenesis, and public health. To quote from Dr. Francis, epidemiology extends the boundaries of the universe in which disease can be viewed beyond the ward or clinic. It provides a third dimension to the understanding of disease by creating awareness of the nature of the environment in which the disability arises, of the factors in the community which contribute to its causation, and in turn its effects upon the community. So why is it essential? There's many reasons. First, we need to understand the range of variation found among humans. Microbiota probably have similar functions among all humans, but we already know that the species present forming these functions can vary widely. So understanding the variation is critical for planning interventions at the individual or population level. For example, this study showed that intestinal microbiota influences the efficacy of immune checkpoint therapies in cancer patients. These therapies block programmed cell death protein 1, PD1, which unleash adaptive immune responses to cancer cells. While studies in animals and humans demonstrate that bacteria mediate responsiveness to anti-programmed cell death protein therapies, which bacteria are responsible is unclear, and different studies have pointed to different species. It may be that all of the identified species have the same function, but their prevalence varies in different populations, or their expression is mediated by other members of the microbiota, or that other bacteria, virus, fungi, or archaea are responsible, and the association with a specific bacteria is because that bacteria is strongly associated with the presence of these other organisms. It's also possible that our experiments, methods, or analyses are flawed. Second, which microbes are present and whether they cause disease depends on human genetic lineage, behavior, and the local environment. To give an infectious disease example, we know that there are different tuberculosis lineages that correspond to different geographic areas. This graph shows results of a study conducted in San Francisco, and the rate of secondary cases by origin of the individual. And the rate of secondary cases is higher among those of the same origin as their TB lineage. I believe that commensal microbiota are also adapted to human lineages and that they become exquisitely adapted to their human host over the life course. Third, knowing the range of variation gives us a gauge as to what differences in microbiota are meaningful. If 2% of variation is attributed to technical variation, or within-person variation, or between-person variation, identifying a statistically significant difference of 2% between those with and without disease is probably not biologically meaningful. In short, we need to understand the range of variation so we can take it into account as we move towards strategies to use microbiota for clinical or public health interventions. In addition to characterizing microbiota by person, place, and time, epidemiologic studies can generate hypotheses about what the microbiota does. The microbiota might enhance or reduce the effects of exposures on the host. For example, microbiota in our nose and throat might mediate risk of respiratory infection. In that case, manipulating the microbiota might be a way to reduce susceptibility and disease spread. It is also possible that an exposure acts on the host and then the microbiota respond. In that case, the microbiota might be a diagnostic or prognostic. In the natural history of the disease, both might be true, with first the microbiota responding to exposure and then to the host. So I've described the microbiome as a dynamic ecological community, but have shown it as a black box. How we think about the microbiome matters because our conceptualization can drive our research questions. Now, this picture from a recent article by Solis and Baumer shows there are multiple conceptualizations of the microbiome. Microbes stimulate the immune system to tolerate their presence and enhance attacks on invaders, pathogens, making the microbiome seem like part of the immune system. However, we may be more interested in the microbiome functions as they relate to other aspects of human health. Key functions relate to digestion and absorption of nutrients, which makes the microbiome seem more like an organ. Conceptualizing the microbiome as an ecosystem is helpful in that we can draw on ecological theory to form hypotheses and analytical approaches, such as niche preemption and modification, which help explain colonization or pathogen resistance. Pathogen resistance is the one function of microbiota that is essential for life. And for the remainder of my talk, I will focus on pathogen resistance. Pathogen resistance can be directed against any members of the microbial world. And today I'll focus on the potential role the microbiome may play in resisting viral infection. In particular, I'm going to summarize what we know, or what we think we know, from in vitro and animal models of viral bacteriome interactions. I will then discuss some epidemiologic insights into human virus bacteriome interactions using influenza virus as an example. My interest in the role of the microbiome in resisting viral infection is long-standing and results primarily from observations I've made as a parent of 3 children who are now all grown. So to motivate the rest of my talks, I'm going to share one of those observations with you. Many years ago, when my children were little, I was invited to a small meeting at the Grand Floridian Hotel in Disney World, shown here. My host made the kind offer of either paying for a single first-class round-trip ticket or paying for the equivalent, which was enough to take my entire family. And we enjoyed the swimming pool, the Mickey Mouse bathrobes, and the fireworks. But about halfway through the trip, my middle son developed pink eye. And as he put it in his vacation report, pink eye is gross. And shortly thereafter, my oldest son and I got sick. While my middle son had sort of a classic mild pink eye, which is gross, my oldest son had light sensitivity and pain, and I was physically entirely sick and had exfoliating cells from my eyeballs and was still wearing sunglasses indoors when I was back at work 2 weeks later. Notably, my husband and my youngest son never got any symptoms. So previously, we might have attributed these different clinical presentations to host factors like age, underlying immune system, or chronic disease, or history of prior infection, or to genetic factors like polymorphisms and immune genes, or agent factors like pathogenicity, virulence, and dose. However, there is increasing evidence that the microbiome also plays a role. Although the virome and bacteriophage likely also interact in important ways in the microbiome, my focus today really is on eukaryotic viruses, so viruses that infect human cells. The microbiome may resist viral infection or it may endorse it. And this may be a property that emerges from interactions among the microbes present or from the presence of one or a few microbes. Although the microbiota consists of bacteria, virus, fungi, and archaea, our understanding of these processes are limited primarily to the actions of bacteria. Much of what we've learned has been gained from in vitro and animal models. However, generalizing from these models to humans can be problematic. A particular concern is animal models and even human experimental models do not represent the range of variability in the human microbiome, as I mentioned earlier. And further, without conducting human studies, we cannot tease out the effects of the microbiome on susceptibility to viral infection, disease presentation, and duration of infection from other known risk factors. Understanding the interactions between human viral infections and the microbiome is important. We all know we are facing a pandemic by a virus and as yet have very few effective treatments. We also do not have good predictors of who will have mild or no symptoms or end up in the hospital. I believe the human microbiome might explain some of this variation in virulence. If there are microbiota that are associated with decreased risk of infection or shorter duration of symptoms, that would provide some avenues for potential prevention or treatment strategies. In this slide, I'm showing you that viral infections are very common. We have influenza epidemics every year, which cause the deaths of an average of 30,000 Americans, but other viruses cause serious illness. We're increasingly aware that a portion of most viruses cause no symptoms. For example, these are results from a study called the Utah Big Love Study. It was a one-year prospective investigation of 26 households that totaled 105 individuals that was conducted over a one-year period, which included the second wave of the 2009 H1N1 influenza pandemic. They took weekly NAIR samples regardless of symptoms. And participants had a mean of 7.3 viral episodes per year, which varied by age. Children younger than 5 years had more viral detection episodes at 12.1, and older adults older than 40 had 3.3. Particularly notable is the relatively large proportion of asymptomatic cases for common respiratory viruses. So 46% for rhinovirus, 39% for coronavirus OC43, which is a coronavirus that is well known to be adapted to humans, and 34% for influenza A. So pathogen resistance is a well-known and appreciated feature of microbiome. And much of this work has been with regard to resisting bacterial pathogens. However, studies on enteric and respiratory viral pathogens have identified several ways that the microbiome can directly resist viral infection. Bacterial products can kill the virus or can inhibit attachment of virus to cells so they cannot gain entry. Alternatively, the bacteriome can indirectly resist viral infection by stimulating innate immunity, changing the host cell to resist viral entry, or producing products that are internalized by the host cell to degrade the virus. Bacteria can also endorse viral infection. Eukaryotic virus form complexes with some bacteria. These complexes are mutually beneficial, increasing opportunities to bind to host surfaces. When bound to bacteria, the virus can move in between human cells. Direct binding to bacteria followed by internalization into the host cell could allow multiple viruses to affect the same host cell, promoting viral recombination and generation of progeny virus. Virus can also upregulate host cell surfaces to which bacteria bind and downregulate bacterial genes associated with a colonizer state. Bacterial products can also regulate immune tolerance to the virus, enhance virion stability, and promote infectivity. This beautiful picture by Rao et al. Shows a complex of influenza A virus with Streptococcus pneumoniae, which is shown in green, and influenza in red. While proof of physical interaction is new, the observation that pneumonia caused by strep pneumonia often follows influenza infection is well known. Strep pneumo usually is found in diploid form in pairs, but may form chains as shown here. And if you're wondering why the influenza doesn't look like little circles, it's because clinical isolates of influenza virus produce long pleomorphic virus particles, which can be extremely long filamentous virions. Influenza strains that have been adapted to laboratory growth typically produce only the spherical virions, as shown as the next slide, which is what I thought they only did. But in fact, if you see in the upper right A, influenza virus can produce these long filamentous particles. So we know that some bacterial viral interactions are positive and others are negative for the virus, that some bacterial viral interactions are positive and others are negative for the bacteria. The interactions may be direct or indirect via effects of the host, and observed interactions may be attributable to specific taxa or properties that emerge from the microbiome structure and composition. So this is all very exciting. But can we observe these interactions in human populations in epidemiologic studies? So I'm sure you all know we can detect the presence of virus when new viral particles are released, and we can test for antibodies to detect prior or latent infection. And this image just to show you can see on the right the viral particles being released, it also shows that antibodies can block influenza from attaching to cells, entering cells, and prevent the release of influenza virions from the infected cell surface. Using genetic sequencing, we can detect what taxa are present in the microbiome. With metagenomics, the microbiome genetic potential. Transcriptomics, we can detect what functions are ongoing, and using metabolomics, the products of host microbiome and microbial interactions. Amplicon sequencing is most commonly used in epidemiologic studies like the ones I'll present today because it's the least expensive and there are well-developed bioinformatic pipelines that are relatively user-friendly. Amplicon sequencing entails sequencing all or part of a gene, and most often that gene is a highly conserved gene, such as the gene that codes for the ribosome. So 16S rRNA is very typical. All cells have ribosomes, and there are also well-curated databases that enable mapping the gene sequence of the 16S rRNA to specific taxa. As we do not have complete libraries of all microbial sequences, the data can also be analyzed as operational taxonomic units, which you might see OTU, which assume that changes in the genetic sequence correspond to different genus and species. we decided to explore whether we could observe viral bacteriome interactions in the human system using influenza virus. And influenza virus is an excellent system because it has epidemics that occur annually and affect large number of people, as seen from the summary of the 2019-2020 season in the US. Infection results in a range of severity from asymptomatic to life-threatening, and its life strategy is to rapidly move from host to host as infection stimulates immunity, which means it needs large populations in relatively close contact to maintain itself. Its other strategy is to rapidly mutate so that despite a host being immune to one strain, it's susceptible to another. And importantly, there are known interactions between the nose-throat microbiome and influenza infection. Influenza often leads to secondary bacterial infection, particularly pneumonia, and much of the in vitro and animal model work on viral microbiome interactions are in the context of influenza. Further, the incidence, severity, and associated bacterial pathogens differ between epidemics, as shown here. At the time I was thinking about how to conduct this study, I had the good fortune that Aubrey Gordon joined our faculty. She has ongoing household influenza transmission studies in Nicaragua, including storage samples from a previous study period. And also fortuitously, Q Han Lee joined my group as a graduate student, and he was very interested in the problem, and Q led the study, which was the topic of his dissertation. The study was conducted in District 2 of Managua. The population is mostly low income, but varies widely, and there are large households with extended relatives living in the same house. And the population pyramid does not look anything like the US pyramid because about 50% of the population is under 25 years of age, and younger children are more susceptible. So this is the study setup. Aubrey had us set up surveillance at clinics for individuals with influenza-like symptoms. If they actually tested positive using an influenza rapid test, they and their household were recruited for study that day. Nose, throat swabs were collected from index cases and household members at time of enrollment, and every 2 to 3 days for about 2 weeks. So we know household members are exposed, and we know whether they had influenza. And we also know whether they acquired influenza during follow-up, and if they did, we know their symptoms and when they started shedding virus. We used an aluminum MIC system with a validated dual indexing method to sequence the V4 region of the 16S RNA gene, and this was done by the microbiome laboratory here. And we also did the sample when influenza was first detected and at end of follow-up. Then we aligned and quality filtered using mother and oligotyping to assign reads to taxonomic units using HOMID database. We then reused Dirichlet multinomial mix models from all sequence samples to identify community types. To assign taxonomy, we searched representative sequences of each oligotype against the human oral microbiome database. So we use these data to test these hypotheses that microbiota mediates susceptibility to influenza viruses, that microbiota mediates time of symptom onset post-influenza infection, that microbiota mediates duration of viral shedding, that these effects might be attributable to individual taxa or microbiome structure and composition, and these effects might vary by influenza type. We identified 5 community types and then use those community types to assess characteristics of the samples. The community types differed in alpha and beta diversity, and what's shown here are violin plots that with a captured box plot with the shape of the distribution around it. So note that community type 5 had the lowest alpha diversity, which captures the number of species and their relative abundance, and the highest beta diversity, which assesses the heterogeneity of species found within the group. Both high and low alpha diversity have been associated with resistance of microbiota to invasion. As expected, the infection rate among susceptible household contexts was highest among children aged 0 to 5, shown in green. However, the secondary attack rates also varied by community type. The pink bar shows the attack rate for all ages combined, and the number on top of the bars is the sample size. The lowest secondary attack rates occurred in community types 4 and 5. Interestingly, none of the 83 individuals with community type 4 were ever infected with H3N2, which was the predominantly circulating influenza type during the study period. And also notably, very few children aged 0 to 5 had community type 4. This is a complicated slide, so let me take a minute to orient you. This shows the results of using an individual-based hazard household transmission model to adjust for age, influenza vaccine status, household size, by influenza type. So on top are the associations of each of those community state types with influenza A, H3N2. On the y-axis is the relative susceptibility, and on the x-axis you show each relative shows it each community state type, and so each community state type is compared to all others. So you'll note in the first column it says the referent community state type 1. You can see community state type 1 compared to community state type 1, community state type 5 was much less susceptible. And this was true regardless of what community state type you compared it to, except of course when you compare it to itself, this relative susceptibility is 1. So it looked like community state type, this on top, I'm sorry, on top this is community state type 4, you see lower susceptibility to infection regardless who you compare it to, with a range of 0.07 to 0.12 with reference to all the other community state types. Similarly, for influenza B, we see that community state type 5 had the lowest relative susceptibility compared to all other community state types. As a sensitivity analysis, we refitted the model using symptomatic influenza infection, and the results were similar. We used the same model to assess associations with oligotypes, and we chose these oligotypes because they were the major contributors to the community state type distributions. This is a forest plot which compares the relative susceptibility associated with each taxa. I don't expect you to read it all, but I want you to focus on the boxed ones, which highlight the difference in relative susceptibility by taxa for influenza A and influenza B. And what's notable in the ones I've highlighted is that the relative susceptibility was different for these same taxa depending whether it was influenza A and influenza B. And so some taxa were positively associated with decreased susceptibility to A and associated with increased susceptibility to influenza B, and vice versa. We also looked at propensity to transmit to others by measuring duration of viral shedding. So the longer the virus is shed, the greater the chance the virus is, of course, going to shed to others. So if we knew how to cultivate the right microbiota, we might decrease spread of influenza and other viral infections and reduce the number of secondary bacterial infections. In our analysis, community type 5 was associated with a shorter duration of shedding, and it was also associated with a longer time to infection, that's the serial interval, and longer time to shedding onset after adjusting for age, sex, smoker in the household, household crowding, clustering by household. And just a reminder, community type 5 was the least diverse. in terms of the number of species present and the relative abundance. So what do we learn? Well, we learned that the nose-throat microbiome is associated with susceptibility to influenza infection following exposure, with time to symptom onset, duration of symptoms, duration of viral shedding once infected, and serial interval, and that these associations depended on influenza viral type. Like most epidemiologic studies, we didn't really answer as many questions as we raised. So there's a question of, might these apparent protective associations reflect prior infection? It's possible that the microbiome changes in response, so microbiome really wasn't responsible for what we saw. The microbiome, if the microbiome is responsible, are these effects attributable to individual microbes or interactions among microbes, or interactions of individual or groups of microbes with the host? And we only measured influenza virus and the bacteriome. So it could be that the factors that we saw were related to something we didn't measure altogether, like phage or fungi, other virus, or host genetic factors. So we have a lot more to learn. I'd like to close with a few words about why understanding the microbiome is important for public health. Public health is about preventing disease and disease progression. With respect to influenza, it might be possible to cultivate beneficial microbes to decrease susceptibility to initial infection by enhancing our immune response. We know that viral infection disrupts our microbiota, so we might be able to restore microbial communities, and that might decrease risk of secondary bacterial infection like pneumonia and ear infections that frequently follow influenza infection. And finally, we might be able to manage our microbiota to enhance resistance to influenza invasion. I would not like to leave you with the idea that the microbiome are our friends. Each microbe is in it for themselves, and individually, they just want what we want. They want to have a home, food, and raise a family. And if it's to their benefit to collaborate with a pathogen, they will. And if it is to their benefit to live peacefully with their host, us, they will do that. So it's to our mutual benefit to figure out how to become allies with our microbiome and to work with our microbiota to help them help us to be protected from pathogens. So thank you for your attention, and I look forward to further discussion.
Thank you so much, Betsy. That was a really great presentation and really helpful for beginners, I think, as I'm new to microbiome research, and I was able to follow very well. So thank you for that. So if anybody has questions, please post it to the Q&A, and we will pose those questions to Betsy. So I guess I will start off with the question while other people are typing in. So usually I thought people think of higher diversity levels as being better, being healthier. However, in your study, you showed that the lower diversity level was actually protective. So I was wondering, is this an exception to the rule, or are there situations where this is common?
So that's a real, so Ashley Shade has a sort of a interesting article called like diversity is not the answer, it's the question, which I might refer you to. But the fact is diversity is sort of an emergent property of the microbiome. So if like you think about different agricultural communities or different plant life, it might be easy to think about. So like a jungle is extremely diverse. It's extremely diverse. And yet if we do slash and burn, you know, it's not very resilient. If you think about a monoculture, it can be extremely effective at resisting disease until the right disease comes along and it can be wiped out. So, you know, we know diversity overall is a good thing, but there are some advantages in each, and you can have very stable systems that have very different characteristics. You know, so it depends on the other environmental circumstances. We know during pregnancy, for example, that the vaginal microbiome becomes less diverse over the course of pregnancy. It becomes enriched for lactobacillus, which must be very protective because it's something that sort of seems to, as best as we know, be very universal. It's an acid producer, so perhaps it decreases risk of infection, but also might seed the gut microbiome of the infant, which through vaginal delivery. So there's just so much we don't know. Yeah. So I see there's a question. Laura asked, with respect to COVID-19, there are striking disparities between African Americans and non-Hispanic white populations. There are differences that could be due to social factors, other studies of stress in the microbiome. Well, we certainly know that there's a host microbiome sort of like mental interaction thing going on. Most of this work has been, you may have seen like with that your microbiome tells you that you need to eat, but we know that certainly your microbiome does react, I think, to stress. So that seems like that would be an important area to study. I haven't followed it, but I would be certain that it's true that there are interactions. So if you think about it, if you think about like when you sweat because you're nervous, and you think about when you sweat because you're doing physical exercise, there's a difference in odor because there's a difference in what you're excreting and what bacteria that it's selecting for. And that's so whether but we have to be cautious because we don't know whether the microbiome are actually just reacting to these changes or they're actually part of the causal pathway, and it's possible that it could be both.
We have another question from Jonathan Kim. So you talked about how we have different microbiotas in different areas of our bodies developed by the niche they hold. Does the environment alone prevent the microbiota from migrating or growing in other areas? Are there instances where different microbiota communicate with each other or different systems, or a single universal microbe that talks with itself, being in both, for example, the nose, mouth, and skin of the face?
So that's a great question. So there is local selection, and then there is selection by the other microbes that are present. And it seems that there's quite a bit of biogeography even in the small areas and strain variation. There was a real, I'm trying to remember who did it, but we saw a real interesting talk where someone actually looked at the biogeography on the face and found different strains in terms of like different hair follicles on your face, even though they were like the same species. So one thing to know about bacteria is that bacteria are populations, and so we think, oh well, you know, it's Staph aureus, and the answer is, well, actually they are much more genetically diverse than all the human population are from each other. So there's quite a bit of strain variation, and they can engage in horizontal gene transfer, so that can make them seem very different. They can acquire virulence factors and like antibiotic resistance from each other. So we do know that different microbiota do communicate with each other and with different systems. If you're thinking about different systems like different types of microbes, there are synergies and anergies between different microbes. So I showed you how like strep pneumo and influenza can work together to help each other invade the host. Remember, strep pneumo is normally a commensal in the human host. So that's an interesting thing that's changing there. But we know that Candida yeast can either be a friend to some microbes and not friend to others. So there's not like there are microbes that we find on many places of the body that can be all related to each other, but there's going to be some population variation even within them.
Okay, we have another question from Thu Lee. He asks, as the host's genetic background is a potential confounder in the influenza study, did you look at relationships between microbiome and influenza among people in the same household who would have similar genetic background? Would the microbiome in the same household be varied enough for this analysis?
That's a great question. Yeah, if you look at the strain level, even people within a household are more similar to each other than they are to like other households, but they're still quite individual, so you can tell the differences in their microbiome. But this, looking at genetic background is something that I'm very interested in, and we have some ongoing studies where we actually have the human genotypes relative to the microbiome characteristics. And so that's something that is analytically challenging, but is a real interesting question. It's complicated with an infectious disease, however, with influenza, because you have behavioral factors that make a difference in terms of exposure, right? So how close you're to a person, whether they shared a bed or not. We did look at crowding. We looked at crowding to see if that made a difference. And we grouped the microbiome, as you can see, though we did look at individual species, we didn't have a strain variation. And we reduced the microbiome problem by looking at these community state types and still got a signal. But we really don't know what's going on in there. It's a great idea, and it's something that we really would like to look at in more detail, looking at the range in genetic background. Because remember, even people in the same household who share parents are not genetically identical either, unless they're identical twins.
So we have another comment from Jonathan Kim, who asked a previous question about microbiota communicating with each other from different systems. He says, I was thinking of different systems like the central nervous system, which tries to keep itself sanitary, yet can respond to signals being sent from the gut.
Yeah, so let's see. So we do know that microbes do communicate with each other. But I think you're going to have to, maybe we can unmute you so we can get, you can expand on that, because I'm not exactly sure what you're asking. Can we unmute him? I don't know how to do that.
Can you hear me?
Yes, great.
Hi, Dr. Foxman. I guess my question was kind of following up on a former colleague I had who worked at the Irish Institute for sort of microbiota, or the microbe. And so their studies were heavily focused on sort of the gut bacteria and its influence on the nervous system, and how it can, not specifically colonizing the nervous system itself, but in its byproducts, kind of being used as neurotransmitters to change, say, mood or different behavior outputs.
And so you were wondering whether, so yeah, that's a very interesting thing, but you were wondering like if, so what's the actual question though? That's it.
I guess I was just curious of is that very common to where, say, a system such as the nervous system or places that don't have a microbiota but are heavily influenced or get communication or talk to places that do have microbiota?
So we do know that the microbiota is communicating to all the host cells wherever it is, because to induce immune tolerance. So there's definitely ongoing communication between our host cells and our microbiome, and other products can make a difference. And the gut's particularly interesting because this issue of the leaky gut and the microbial products, which can influence you, that way. But microbes, they live in these, they have the ability to talk to each other through chemical signals called Quarren sensing, which allow them to change their functions and talk to each other, like build things, disperse, and so forth. So these signals, actually, like bacteriocins are kind of like a bacterial product that's like an antibiotic, which they can use for communication and also to kill other bacteria that they don't like. And some of the bacterial products are similar to some human antibacterial products. So there is a lot of communication going on. The gut nerve system is a very interesting one. I'm presuming that goes on in other ways too, but I'm not familiar with that.
Okay, so it's not, it's more of a localized communication, not sort of a long distance or using, say, the circulation system.
Well, the thing about the gut is that you get absorbed into your blood, and that makes it be long term. But that can happen probably in your mouth too, and other places where there's close communication, like the capillaries around your salivary glands. That might be another place where there's communication, but I'm not aware of that, other than the one that seems to be best understood or most explored. I don't know if it's the best understood, but most explored is the one that you mentioned. But I haven't heard other ones. But I would not be surprised if that happened. We just don't know. Good question.
Thank you. Thanks for your answer.
So we have another question from Nisha De Silva. She asked, what role do antimicrobial peptides have in the variation and the microbiota at different sites, both in the same individual or between individuals?
So, Nisha, are you talking about antimicrobial peptides produced by the human host or produced by the individual, by the microbes?
Nisha, I think I've allowed you to talk.
Let's see. She's still muted though.
Oh.
Okay, Nisha, go.
Produced by the human host.
Oh, okay, by the human host?
Yeah.
Yeah. I don't know if anybody's really explored that, but I'm sure it makes a difference. Because we have. I mean, I think about it as it's why I like the sort of treaty analogy. I mean, we sort of have a treaty with our microbes, but the treaty is, like respect but verify, whatever it is, so that you have things to kind of keep them in check. Because we know that commensal microbes can also attack us, and they will when we're dead. They will too. That's one thing that happens when you die, is your microbes suddenly are out of control. I think that would be something that would be interesting to explore, because a lot of that is very local. It's not like that we have antimicrobial peptides all over the body when you have a local infection, right?
Thank you.
So does anyone have any last questions? Please feel free to submit them to the Q&A. We will be wrapping up in just a minute. I'd like to thank everyone again for joining also.
I have another question, if you don't mind. Hello?
Okay, go ahead.
When I think of microbes, I also often think of the mitochondria and how it's kind of evolved to be very sort of interdependent on our own host cells.
Right.
The microbiota you've mentioned is kind of looking out for itself, very independent, but do we see a lot of cases of kind of this convergent evolution of it being very dependent on our own on our host cells?
Sure, absolutely. I mean, you know, we have commensals that can only live in human hosts, right? So they're very well adapted, like Haemophilus influenza. It's an obligate bacteria for humans. And they have to figure out what to do to live there, right? And and if you look at your gut microbiome, that's another place where they're clearly, that's a co-evolutionary thing. We like starting from when you're born and when you breastfeed, like breast milk selects for particular microbes to help start building a healthy microbiome. So that's a co-beneficial thing. But the microbes that we get, some of them we get from our mother, but we get most of them from the environment. And the estimates of how much we get, how much we're sort of like initially colonized with, and how much is sort of generally obtained, so sort of horizontally versus vertically transmitted, there's guesses is that we actually inherit less than 10%. So there's got to be selection by the host for these particular ones, and there are lots of microbes that it's a mutual admiration society, but still, under the right circumstances or the wrong circumstances for us, they will take advantage of other things. So one system that I know well is like commensals that live in your nose throat. If you have a viral infection, they might think, oh, I can go up that eustachian tube and I can get in the ears. You know, that's a great place to be. There's hardly anything in there. It's a whole new territory. But it's a trap for them, right? And it makes you miserable. You know, they can live there, but you know they can't get in. If they need to get into another host, they can't get there. Eventually, all microbes, you know, we may have the same microbe with us for our entire life. We probably all do, unless we. And some of them we may pass on, and some of them we may not. But eventually, they have to get into another host one way or another, but they can be highly co-evolved with us. Does that answer your question?
Yeah, the sense of there's independent operating microbes, but there's also ones that are kind of adjusting and adapting themselves to be very codependent on our own host cells.
Right. But eventually they do have to figure out what they do when the host dies, right?
Right. No, thank you.
Uh-huh.
Well, thank you again, Dr. Foxman, for this presentation. It was really wonderful. I think that's all the time we have today, so that will conclude the webinar. For those of you who will be joining the post-event discussion, you should have received instructions to join the separate meeting earlier today. If you have any questions, please email me, Katie at KMRENTS@umich.edu. Thank you again.